So it's really a pleasure to have with us John Pandolfi from the University of Queensland, Brisbane, in Australia. He's, well, a paleontologist, a paleoecologist, a marine biologist. You know that he started his research working on the Paleozoic, but then he moved. When he moved to Australia, I think he started to look also at the most recent past and also to modern reefs. So John, please, welcome.
Thank you, Francesca. Thank you to the organizing committee and the scientific committee for inviting me to open this conference. It's very much an honor and I appreciate it. It's lovely to come to Italy where my family ancestry is from. I was born in the United States, but my father was from Ascoli. My father's parents were from Ascoli Piceno in the Marche region, and he met his wife in Ellis Island in New York when they both immigrated to the United States. And they never spoke, I'm sorry? Oh, okay, it just sounded loud to me. They never spoke any English, and my father grew up learning English at the primary school in Wilmington, Delaware. And that was very difficult for him, so he, when he raised his family, he was not going to have them subject to any of that kind of discrimination, so there was no Italian that was spoken in my family. So I'm sorry I can't give you the lecture in Italian today, but I'll give it to you in English. Here we go. I'm gonna speak today about some of the work I've done throughout my career in line with the conference theme, which I very much appreciate. And I'll start off by giving you some insights, although I'm sure you're all very familiar with the global pressures that humans have been introducing in both terrestrial and marine environments from the very first onset of human interaction. We all know about the quaternary extinctions of the megafauna on North America, Australia, and elsewhere. It takes very few humans to be very efficient in decimating large populations of organisms. And this kind of exploitation in the sea and land use changes in the terrestrial habitats, and then the urbanization of those habitats, the coastal development in the oceans, has led to basically where we are today. And in not a very good place. In the marine environment, you can talk a lot about different resources, world fisheries. Today we're actually producing more aquaculture in the seas than we are getting from wild stock. The corals, the loss of corals decline is increasing. The loss of mangroves is increasing. We've got lots of different reasons. And you can put up any kind of different variables here that people study that are leading to the decline that we're seeing in the Earth's biota. Here I've shown turbines and seabed mining, shipping, and dead zones, all sorts of depressing kinds of things happening, which I'm sure you're all aware of. Well, coral reefs are no exception. They are also in rapid decline for lots of different reasons. Coral bleaching, changes in land use leading to habitat degradation. Crown of thorn starfish we have in the Indo-Pacific. Great Barrier Reef right now is going through its fourth wave of crown of thorn starfish decimation alongside all the bleaching. Overexploitation, some top-down effects from overfishing, causing trophic cascades in coral reef environments, and coral diseases are also a primary cause of degradation in coral reefs today. So lots of things to worry about. Some people think, oh, well, let's just start to restore reefs, and while this is a beneficial in many ways, especially in terms of raising awareness of people around the world to what's happening. Unfortunately, even when we look at these large-scale kinds of, you can't really see that, can you? Large-scale sort of efforts to rejuvenate reefs and to plant corals into the reef environment. The effects are generally quite small. This sort of thing, there's a scaling problem here that can only be done over very small spatial scales. Well, what's the answer? And I guess what people are generally thinking about these days is what I call holistic adaptive management strategies. That is, trying to use all the information as possible to formulate management strategies. There's an adage going around that it's not important to manage reefs, it's important to manage people. So how do you manage the behavior? And I think one of the things that people respond to is multidisciplinary research, lots of information gathered in different ways. And there's lots of disciplines that people, valid disciplines that people use: conservation physiology, species on the move, looking at distributions, et cetera, et cetera. But what I'm going to focus on today is what I call conservation paleobiology, or what, not I call, but what is now a fairly established field within paleobiology and paleontology is conservation paleobiology. And this book has a nice definition of it. Dietl and Flessa in 2011 applies the theories and analytical tools of paleontology to the solution of problems concerning the conservation of biodiversity. And that's what it is, but it's important to think about what it isn't. It's not confined to any particular time period. I'm going to bring up this theme time and time again in this talk. It's not confined to any particular taxon or ecosystem, and it's not limited to the study of the fossil record of extant organisms. The remit of conservation paleobiology is much broader and includes all those organisms that some of us study in the ancient past that even have no modern analogs. So what am I going to talk to you about today then? I'm going to provide a series of examples to show you in my work how I've tried to apply these principles of conservation paleobiology to address environmental and ecological challenges in the modern world. I want to show in this talk and reinforce the wonderful work that's being presented here that I'm very excited about because of the theme that Francesca and her colleagues have introduced. And sort of pursuing that theme through systematics, paleoecology, big data, and conservation approaches. And again, any organism, extinct or extant, any time period in any ecosystem falls into this rubric. So now I'm going to present to you three sort of research vignettes that I hope will illustrate some of these issues. Okay, the three vignettes are, the first one is a paleoecological approach to looking at coral reefs and how we might go about defining natural baselines in the sea. Some of the lessons that we'll get from that is trying to know what to conserve, what should managers focus on, and also identifying drivers of ecological change. What are the drivers of the change? The second vignette will look at the causes and consequences of ecological novelty, which I'll define for you. We're going to look at some of the demographic drivers of ecological novelty and biodiversity loss, and sort of look and see what that analysis says about the approach we might take to conserving novel communities or not, because some people believe that we shouldn't. And then in the third vignette, we'll look at fossils and climate change, and mainly looking at species distributions and some of the traits that are important or that respond to climate change. Okay, so the first one, paleoecology and defining natural baselines. We're going to look at a couple of studies of community persistence over multiple scales in both the Caribbean and the Indo-Pacific. Again, the lessons are knowing what to conserve and identifying drivers of ecological change. And I'll just spend one slide looking at something I'm excited about in deep time as well. Okay, so this is a graph from Joe Connell in 1997, who, along with Carden Wallace and others who's in this room and others, had been looking at quadrats at Heron Island on the Great Barrier Reef, going back, I think, every year for 30 years. And what Joe and his colleagues found was very much a pattern of disequilibrium in percent coral cover. Some years the coral cover was high, some years the coral cover was low. And I think this led to an understanding that in reef systems, species composition varies very widely. And they're disturbance driven. The clock was constantly being reset by cyclones, and they were never reaching an equilibrium state. And so I decided to go out and sort of have a look at this at a bit different scale on the Huon Peninsula. So I looked at these terraces on the Huon Peninsula in Papua New Guinea. The locals call these the giants' steps. And we all learn in geology that the oldest rocks are on the bottom and the youngest at the top. Well, because of the tectonics of this region, the youngest rocks are at the bottom and the oldest rocks are at the top. And we can basically sort of, the formation of the terraces occurred through the interaction of quaternary sea level changes and tectonic uplifts. So you can basically reset the tape every 10,000 years or so and look at a new coral reef community and ask the question, how similar is this new community to the ones in the past? And I did that over 115,000 year time scale in Papua New Guinea. And so each one of these numbers represents a particular reef terrace at a particular time in a particular place. And I had 3 sites along this 35 kilometers of coastline. And so what you see here is that this is an ordination, and communities, coral communities that are closest to one another have the most similar composition, and communities that are the most further away in the ordination have community compositions that are very different from one another. And basically, the ordination shows a shotgun. And while there's a lot of variability within and between terraces, there's really no sort of structure. There's no real differences going from one time frame to another. So species composition persists through multiple reef building episodes in this system. So I wrote a paper where I called this limited membership in coral community composition. Now, this paper was based on presence-absence data, because you can imagine in the hot sun in Papua New Guinea with a species diversity of about 400 coral species, that it would have been very difficult to go down to the species level. So to do abundance data, and so this was just presence-absence. But in Barbados, I was able to repeat this study at the species level and using abundance data. So we were able to run transects along specific terraces. Again, Barbados is characterized by a series of terraces on the western side and the southern side. And again, it coincidentally turned out to be a 115,000 year interval of study in Barbados, but 4 different time periods. And again, when you do the ordination, the coral composition, another shotgun sort of pattern that shows, sure, there's lots of variability within and between time periods, but there's no real structure to the where time periods are actually giving very different compositions. So again, species composition is persistent through multiple reef building episodes, and the reefs are displaying this limited membership in community composition. Very different story than what we found in the what Joe Connell and his colleagues found over much smaller spatial and temporal scales. If we look at the distance between communities along the coastline and look at their similarity, under a model disequilibrium or a model, a neutral model, one might expect that the similarity would decay through time. So these, you'd expect lines that go down like that. But in fact, it doesn't matter whether the communities were 200 meters away from each other or 30 kilometers away from each other. The similarity among them was pretty consistent. So again, that similar community structure being repeated not only in time, but in space. Now, the interesting thing is that if we sort of go almost to the conservation side of this thing, and this is our ordination again in Barbados with the 4 different time periods. And then we add in the composition of the modern reefs in Barbados. Something very, very dramatic happens. All these points in the Pleistocene basically drop down into almost a single point, and really very, very distinctive and very, very different from the modern reefs in Barbados. And this is because those modern reefs, they look substantially different from the Pleistocene because they've lost their original sort of community structure, lost the acroporas, the large branching corals are gone, or they're very, very much reduced. The whole structure of the community is very, very different, okay? And this is a typical scenario. If you ask the question, how typical is the Barbados result across the Caribbean? Well, you can go to this graph, and this is basically 2 graphs. This is one here for Palmata and one here for Cervicornis. And this graph applies to the reef crest zone in the Caribbean from about 1 to 5 meters that used to be dominated by Acropora palmata. And this graph applies to the 4 reef zone of Acropora cervicornis in the 4 reef zone. And this is a literature survey, so it's big data as opposed to collections, and it just gives the percent of Caribbean sites that were dominated by the respective species of Acropora in the respective reef environment. And you can see in the Pleistocene and the Holocene that the numbers were quite high for each zone, that Acropora dominated each zone, just as it used to in the 1960s and 70s in a lot of places that people would go and dive, Discovery Bay, for example, in Jamaica. Well, if we go to the pre-1980s, we see a substantial drop in the percentage of Caribbean sites where Acropora is dominant for both of these zones. And so things were happening between the Holocene and 1980, and I've written other papers that have gone through that time period between the Holocene and the modern during different anthropological stages where we've documented some of the things that have happened. So this wasn't surprising. But then what happens is post-1980, we get another significant drop. And of course, the drop post-1980s was due to the loss in diadema in 1982-83. As it turned out, it was the last remaining herbivore on the reefs. And when those herbivores were gone and the corals died of natural attrition, they were not able to recruit back onto those substrates because the macroalgae prevented them from doing so. So we got a big phase shift or regime shift in the 1980s. Now, what's interesting here is that when I show this to my students, I show them this particular photograph, and I say, look, isn't that a beautiful reef? And they all shake their heads and say, yes, it's a beautiful reef. But it isn't. It's beautiful coral, but what you don't see in this picture is any fish. And that's because all those herbivorous fish had been overfished and taken out. And so by the time we got this disease and diadema, that was the straw that broke the camel's back, right? Well, we can shift over to the Indo-Pacific, and we can find some similar sorts of things happening. This is a study a student of mine has completed, George Rolfe, and it's basically we went to some reefs in the GBR and looked at their history over the past 1,000 years using reef sediment cores, looking at coral composition. We're able to date the corals very, very precisely using uranium, thorium, radiometric age dating. And we've done that both with cores and also with death assemblages that were just lying on the sea floor. And so what happens, the results here are a substantial decline in Acropora on the living reefs at sites A and C, which is the blue and the red, and a rise in dominance of Pavona at site B, which is the green. So what happens in these death assemblages is that all the Acroporas are dating to about the mid-1930s to 1940s. To 1950s, but we don't get any younger Acroporas, or very few. There's one there, but in the for the Pavona, we get very much younger dates. So that Pavona community is just it's a death assemblage that's dominated by a normal turnover of Pavona, which is, I'm sorry I don't have pictures here, I should have, but Pavona is a sort of a weedier kind of coral than Acropora. And then if you go to the cores, we find at two of the sites, Acropora, which is in red, has dominated the cores all the way up to the present. So at these two sites, the loss in Acropora that occurred around 1940 to 1950 is very, very different from what the reef used to look like, because it used to be dominated by Acropora. The middle site, where Pavona is, live Pavona is occurring, we had Acropora all the way up to the 1930s and 1940s in the cores, and then it was replaced by Pavona, okay? So this transition that we see in the death assemblages, we can also see in the cores. That's one of the times where you sort of fall off your chair in your office because you think, no, this can't be true, right? These two different data sets can't show the exact same thing. But in fact, they did. So a real problem here with Acropora not being able to some of my colleagues like to say, oh, well, what killed the corals? I don't care what killed the corals, although, well, I do care because it was land use changes. But the really important thing is, why aren't they coming back? Why aren't they recruiting back onto those reefs? And that's the issue. And the reason is shown in this photograph here. This is the Burdekin River that comes out around near Townsville. When it floods, a lot of the years it's just bone dry. When it floods, it brings out all this sediment onto the reef. This is the Palm Islands where this study was completed that I just showed you. And you can see it entirely bathed in sediments and runoff from the land. And in fact, these are the sorts of things that have been driven by land use changes ever since European colonization in the 1850s, 1860s in the Queensland coastline. So these are some of the issues that are driving these patterns. So what are the lessons from this study? Well, some people suggested that we really don't know what to conserve because coral communities are unpredictable. Look at Connell's work, look at other work. And so how can we sort of go about trying to conserve these ecosystems when, we really don't have a clue on what constitute a natural community. But I think this long-term fossil data provides an alternative perspective that actually says, hang on, we've had this kind of community structure in place for a very long time, and what we're seeing now is very, very different to what we've seen in the past. So we've got persistent community structure in those fossil reefs, and I think we can use that information for implementing and evaluating management actions. Okay, so coral reefs can be managed in the context of a persistent state. It's not all chaotic. There's significant and important differences between reefs comprised of different corals. There's others who have asked about this classic Caribbean zonation pattern and saying, is it anomalous for, between 1940 and 1980, we really didn't have any, a large, for example, in Jamaica, there were no hurricanes. So perhaps because coral reefs should be disturbance-driven ecosystems, we just didn't have those disturbances, and so that's why we had these monospecific stands of Acropora. But again, we can say no to that. We can say that these mono-dominant stands have been a consistent feature of reefs for really over 500,000 years. Anybody who's been to Barbados or read Ken Mezzalela's thesis would know that these terraces that I've shown you actually go all the way back to 500,000 years, consistently dominated by Acroporas. So, and then the second lesson I think from this is identifying drivers of ecological change. The drivers of acropora decline, they predated climate change and disease and acidification. So we can look at other drivers, habitat destruction, water quality, overexploitation in that journey from reefs that used to look like this to reefs that now look like that. So basically a recommendation is to try to enhance the water quality, try to reduce the habitat destruction. And these are all simple and well-known sort of strategies, but people tend to, we constantly have to bring up these issues and bring up this fight because there's others out there who want to do away with restrictions on coastal development and fishing and things like that, water quality. Now, of course, I haven't even spoken to you about climate change, which is obviously a huge threat, which I hope to get to by the end of the talk, and I'll just have to go a bit quicker. But that's a huge threat as well. Now, I just want to have a shout out that here is a series of raised reef terraces, okay? And it's from the Devonian Basin the Canning Basin, the Devonian Canning Basin in Australia. It's Australia's other Great Barrier Reef, right? And here, instead of the 4 sequences in Barbados or the 9 sequences in Papua New Guinea, we've actually got 75 repeated sequences. I'm really looking forward to this PhD project by Amanda Godbolt, who's getting her PhD at the University of Southern California. And she has decided to tackle this question. This is the Guppy Hill section. She's going to go to a few different sections and do this very similar work of looking for community persistence over time and sort of testing these hypotheses in deep time, where we have environmental information, climate information. So we can do these kinds of studies even in deep time. It's, I guess, what I'm trying to sort of stress here. Okay, so the next vignette has to do with the causes and consequences of ecological novelty. We're going to look at extinction risk in novel communities. First, I'm going to introduce a new framework that I've been developing, and some of the lessons here I'll go through in a minute. Now, there's a paper that I've been very impressed with. Some people laugh at me because I've been too impressed with it, by Dornalis and her colleagues in 2014, where they looked at alpha diversity over time from the 1900s to 2000s, and then beta diversity. And basically, alpha diversity across the global environments, marine and terrestrial, has remained constant through time. But over the last 80 years, beta diversity, or taxonomic turnover, has increased through time, okay? So communities are getting more and more dissimilar to one another. There's more taxonomic turnover in this sort of topsy-turvy world of human degradation. And I like to think of this in terms of novelty, in terms of, what does this mean for the emergence of so-called novel communities? And that's what I'd like to talk to you about for the next few minutes. Well, what are novel ecosystems? For conservation biologists, novel ecosystems are broadly defined as those in which historically unprecedented combinations of species occur, primarily as a result of human influences. You can go to this book here, Hobbes 2013, and find about 10 other definitions of novelty as well. So it's one of those concepts like resilience or stability that people have been sort of floating around and not being too precise about how they're defining it. You can also recognize this concept in the plant paleoecology literature under no analog communities. So no analog communities are compositionally unlike anything found today. Right, okay, what does that mean? So what I've done with my colleagues, Tim Staples, who's a postdoc in my lab, and Wolfgang Kiesling, who is also in the room, we've used a 2-component definition to try to get at this idea of novelty in a bit more quantitative and repeatable way. And there's 2 components to the definition. It's a large change in composition that's faster than a system's normal rate of change. And a novel community must be substantially different from all of its past historical composition. Okay, so these are the two components that we focused on, and we called the first one instantaneous novelty. So this is a time series of microfossils from about 42 million years ago to the present. And it's comparing every time bin, every 100,000 years, with the time bin before it, every time bin with the time bin before it, every time bin with the time bin before it. And where you get large dissimilarity that is higher than a modeled similarity through time, then that's instantaneous novelty. Now the next picture shows what's called cumulative novelty. And you notice here now we go dissimilarity increases upward, whereas here dissimilarity decreases downward, and there's a reason for that. But again, we compare every community here with every other community behind it that went before it. One community here with every community behind it. So I wanted to know, if I flew into the Miocene, how would I recognize a novel community? I wouldn't have anything ahead of the Miocene because it hadn't occurred yet. So the only thing I could do would be to compare my community with everything behind it. So that's what we called cumulative novelty. Now, where instantaneous novelty and cumulative novelty hook up, where you get them in the same time bin, that's what we call true novelty, or true novel communities. Okay, so that was our sort of quantitative approach to try to look at novelty through time. The data set that we decided to cut our teeth on with this approach was the Neptune data set of planktonic communities. So we looked at calcareous nanoplankton, planktonic foraminifera, radiolarians, and diatoms, both by site and also by Longhurst province. Sampled every 100,000 years over 65 million years. Dissimilarity index, genus level analysis, presence absence data. So when you look at that, this is the result. So this is the probability of a novel community for the four different groups. And if you put that together, getting immediate large shifts in community composition occurred between 4 and 7% of the time. And a shift to a new community state that hadn't occurred in the past was about 3 to 5% of the time. So relatively low frequencies of natural novelty in the geological past. And all the rest were so-called background communities, where the community was very similar to the one before it, or was no different from all the ones behind it. And when you sort of splice out or separate out or deconstruct out the instantaneous novelty only from cumulative novelty only from a novel community only, these are the percentages. And again, they're quite low. So one could get an understanding of novelty through time under natural situations. And you can imagine you could bring this forward to time periods when humans were interacting with the environments, and you can see what the great potential can be for such an analysis, and that obviously is the direction that we're going. So this is just a plot of the observed versus expected probability of community state transition, and it's just calculated from the product of the two occurrence probabilities going from one to the other. And all it shows is that most shifts in composition were small shifts. I think we're losing the battery. Oh, no, okay. Were small shifts to non-new states. So the overwhelming majority of transitions were background to background. But to and from novelty were a fair bit rarer. And then going from a novel to a novel state, so all the colors are novelty. When you go from a novel to novel, it's rarer still, but the probabilities are up to 10 times more than expected. So even though we don't get a lot of transition from a novel community to a novel community, even with a small absolute number, the probability is very large. It's a very large probability of going from one kind of novel community to the next. So what that means to us is that novelty essentially begets novelty. If you're a novel community, you stand a good chance of going into another novel community, despite the apparent low probability of novelty in our time series. So there's a heightened propensity for novelty to beget novelty. Right, now I just want to go to some of the drivers of this novelty: extinction, origination, immigration, and emigration, and just show you that the drivers of these states, of these transitions, tend to be a high probability of local extinction. So the shifts to background communities have very low extinction associated with them, whereas the shifts to novelty have very high rates of extinction. And the same thing with origination. Origination is also higher in these novel communities than in the background communities. So the lesson here is that we have these novel to background transitions, and the novel to background is essentially novel community persistence. And they have a lower extinction risk than novel to novel transitions. So bear with me here. If we have a novel community in the modern, how do we want to conserve that? Do we want that novel community do we just want to leave it because it's a novel community, it's not something pristine, or do we want to conserve it? Now, if it's a novel community and it remains the same, it's got a lower potential for extinction, for biodiversity loss, than if it turns into a new novel community. So the idea is that we need to commit triage to novel communities to help them to stay the way they are, even though they're different from the way that we'd like them to be. So we need to facilitate the natural propensity of newly developed novel communities to persist and prevent the transitions to previously unseen ecosystem states that are linked with a heightened extinction risk. Okay, why is that important to this meeting? Who cares? What are you doing talking about forams in a fossil Cnidaria meeting? I'll give you this vignette because contemporary management strategies can benefit from ecological work done on entirely historical assemblages. None of those assemblages that we looked at were modern, okay? But we actually got some pretty interesting conservation messages from it. In order to do that work, we had to have fundamental systematics done. David Lazarus has been working with the Neptune database for decades and has been vetting the systematic underpinnings of that particular database. So that's fundamental to any ecosystem, any study of ecological dynamics, which is in turn the basis for conservation and management. Okay, so it all starts with the systematics. So the age of the study, the type of the organism, whether they're extinct or extant, are all immaterial to utilizing the fossil record for pressing societal and conservation concerns, including climate change. And this is why I was just so happy that Francesca and her colleagues are advancing this theme in this particular meeting, because I think it's very, very relevant. I'm debating now about whether to go into this last vignette because I'm getting, let's just see how we go. Fossils and climate change. I won't tell you what I'm going to tell you, I'm just going to tell you. 2014 to 2017, global thermal stress event, worldwide, maximum heat. We had the incredible global bleaching. Some places bleached 3 years in a row. The Great Barrier Reef bleached up and down the coastline here. 1,156 reefs were surveyed by Terry Hughes and his colleagues, 35% coral mortality, and 22% in the north and central GBR, north and central, and 22% mortality on the GBR wide. The thing came back and bit us again in 2017. Return time for the global bleaching events now is much shorter than the recovery time, which tends to be 10 or 15 years. So the Great Barrier Reef is in a huge decline. What can the corals do? They can move, adapt, or die. Skip over that. corals in the, around the world are moving. This is the study by Yamano in Japan showing the northward march of species along the both coasts of Japan, up to 14 kilometers per year. Those corals are moving. Same thing's been noted in Western Australia, in Florida, and in Eastern Australia. So corals are on the move around the world. So some of that work was the inspiration for Wolfgang Kiesling and myself to have a look at the distribution of modern corals and fossil corals in the Pleistocene. And we chose the 125,000 year interval because it was warmer than the present. Yes, I said that, warmer. So we're going to look at warm Pleistocene and cool modern and look at the distribution patterns. And lo and behold, we find that, that in both the northern hemisphere and in the southern hemisphere, that leading edges and trailing edges of the Pleistocene are displaced poleward during the warm Pleistocene in comparison to the modern. So in the Pleistocene, we've got these species distributions that are marching away from the equator and towards the poles, similar to some of the patterns that are happening today. The other thing is that we looked at the diversity of taxa over latitude, and this is a well-known diversity dip in the recent along the equatorial zone. For recent corals, this happens with lots of different taxa. I can show you a paper by Tittensor in Science where they go through a lot of different marine taxa and show this dip. Well, that's about 10% drop in the modern. In the Pleistocene, we showed that drop to be about 35%. So things are getting too hot in the equatorial zone from -5 to 5 degrees, well, 5 degrees south to 5 degrees north. So that trailing edge is marching away from the equator. So these equatorial gaps were more severe for these Pleistocene corals during warmer climate than the modern corals. So what you predict is equatorial range retractions for future warming conditions. Okay, so let's presume that's going to happen, that species are going to march away from the equator. Well, which species are going to do it? And which traits are characterize those species is the next question that we're asking. And we looked at a few different kinds of taxa, and I put these into, I think, 5 different groups here, 1, 2, 3, 4, 5. Groups 1 and 2 are where we have taxa that range through the equatorial zone, whether they occur in both hemispheres, or whether they occur in one hemisphere and in the equatorial zone, or just the other hemisphere and in the equatorial zone. And then there's taxa that are gappers. They show this equatorial gap, whether they occur in both hemispheres or whether they occur in just one hemisphere or the other. So we can contrast, we can ask the question, what are the characteristics of these range through taxa versus the characteristics of these so-called gapper taxa? So that's what we did. And that was our data set for the modern, and this is our data set for the Pleistocene. And you can see it's much poorer, but in our comparisons, we just sampled in the modern the taxa that we have in the Pleistocene. These next series of slides just hit you on the head with that same sort of gapper versus range through. And because I'm running out of time, we'll just go straight to the results. And we looked at, we had enough information in the coral traits database to actually look at 2 traits, growth form and larval development, whether they were spawners or brooders. And for growth form, we didn't find any pattern among the different, within each group. Within the gappers, there was no difference range through taxa, or with a fossil gapper taxa turned into modern range through taxa. It was all similar to expected. There was no patterns in the growth form. They were spread all over the place. Well, with larval development, the situation was very different. the mode of larval development was associated with species distribution patterns, and I'll show you how. This is a chi-square test of independence showing the Pearson's residuals, and for the growth form, you can see that these values are very small, where we got no significance in the different growth forms with respect to what kind of species distribution pattern. But shown in color is the significant results for larval development. And you can see that the gapper taxa are much more associated with being brooders and not being spawners, whereas the range-through taxa and the taxa that became range-through taxa in the modern, that were gapper in the fossil, show a much a negative trend for brooders, and they're dominated by spawners. So gappiness is associated with spawning corals, whereas range-through taxa is associated with spawners, whereas gappiness is associated with brooders. Okay, so what does that tell us? The mode of larval development, but not growth form, varied among our biogeographic groups. The spawners are less gappy than the brooders, so they're more tolerant of warm equatorial regions. And you think, why would that be, right? Well, it's probably just the result of demographic stochasticity associated with the greater potential for those spawners to have a higher dispersal capacity and colonization potential. So one could imagine that even during the warmer periods, if they were both wiped out equally, as the cooler periods come, the spawners would be better at recolonizing those equatorial regions. So we can't really say that reproductive mode is somehow linked thermally. But they might be associated, the reproductive mode might be associated with other traits that do have some kind of thermal tolerance, but that kind of study awaits further results, I think. So the important result here is that it's more useful for predicting the biogeographic pattern than providing an underlying thermal mechanism for it, which is fine. Prediction's good. Right? Okay, so the conclusions from this whole big talk then is the big data stuff, the Neptune, the PDBB, or however you say that, the looking at quaternary records, that's all good stuff, right? But so are large and small curated collections. They're all valid, important, and needed approaches, I think, to tackle this challenge that we've set for ourselves. So it's essential to incorporate the fossil record at all levels, systematics, ecology, field data, modeling, to address these urgent conservation, ecological, and even evolutionary issues. Nancy Budd and I have published on evolutionary implications of modern and fossil distributions as well. So studying the history of life through time, historical range of variation is really, really critical. What's that history, what that range of variation in the absence of humans, right? And the timing of events give us an idea of the environmental drivers. So geochemical analyses of all of these fossil sequences is absolutely essential as well. And one of the most important things right now, I think, is the prediction of the response to climate change. So lastly, again, any ecosystem, any organism, extant or extinct in any time period, I think we should hold that high as we proceed through the week. I'm very excited to see the talks that have been submitted, and I hope that everybody can sort of somehow think about what they're doing and how that applies to the general theme of the conference. So thank you very much.
I just want to say that this talk of John is recorded, and so we will upload it in the website of the meeting and in the website of the Fossil Cnidaria Association. We have 5 minutes for questions before the coffee break.